Hi, my name is Janelle Larson from the U.S. Geological Survey, and I'm representing a team of scientists aimed at understanding why few lakes in North America have high abundance of amphipod invertebrates. In this presentation, we focus on aquatic plant communities as a factor in their success, but please also see good presentations by Fitzpatrick, Keith, Carleen, and Biegnick as related to this study. Systematic conservation planning is focused on maintaining, protecting, and restoring biodiversity at landscape scales. This model has been used worldwide to identify and prioritize landscape conservation networks. The planners first need to choose which conservation features, like an ecosystem function or a specific species, that's going to be used to represent biodiversity. Biodiversity proxies often include species that are easy to measure, those that are environmentally sensitive, and that their protection will extend to a wide range of co-occurring species. Decade-long debates continue as to whether a single species focus can actually conserve biodiversity. We ask, is the conservation of biodiversity at odds or compatible with the conservation of the abundance of specific species? We've evaluated this question by examining the role of wetland habitat diversity at supporting very abundant populations of amphipods in the Prairie Pothole region of North America. Amphipods are wetland crustaceans that are very charismatic and hence have a lot of nicknames. They're exciting to wetland enthusiasts because they're known as important waterfowl forage and because they're very easy to detect in high densities. However, it remains unclear whether amphipods indicate ecosystem health and whether their conservation focus would extend to lake biodiversity. Several previous studies have shown amphipods have declined across the Prairie Pothole region in the past few decades, which is also likely cascaded to partially cause scaup population declines. Today, amphipods are unevenly distributed across North America. Iowa and Minnesota's wetlands typically have much lower densities than the Dakotas and Canada. Previous research showed amphipods are positively correlated with low fish densities, low hydrologic connectivity that prevents fish passage, good water quality, and abundant submerged aquatic vegetation. This previous work was undoubtedly very informative, but also limited because there were few study wetlands with high amphipod densities. And good news, occasionally lakes still have very high densities of amphipods. We've defined high density as those that range from 500 to more than 7,000 individuals per cubic meter. Basically, you can see thousands of amphipods swimming around in the lake, and you can easily scoop up handfuls. We've proposed studying wetlands with high amphipod densities to refine what environmental conditions are suitable for them. Our specific research questions were, does aquatic vegetation diversity support high densities of amphipods? And what is the typical aquatic vegetation community composition of amphipod-rich wetlands? We've hypothesized that aquatic vegetation diversity would support amphipods directly by providing a continuous, high-quality food base, and coincidentally, because other factors like water quality affect both plant diversity and amphipods. To do so, we studied 49 lakes across many different habitat types of Minnesota. Half of the study wetlands we had known previously had high amphipod densities greater than 500 amphipods per cubic meter. The other half of the study wetlands were selected at random across the larger landscape. These lakes ranged from 1 to 200 acres in size. They were mostly considered shallow lakes, but there were definitely a few deeper lakes as well. Our field surveys had collected information on the amphipods, plant communities, and wetland attributes. Amphipods were collected systematically with a dip net, and all the contents, including amphipods and vegetation, were preserved in ethanol for future lab analyses. We collected plant data at least 50 equally spaced points across the aquatic areas of each lake. At each sampling point, we had collected information on all the emergent, floating, and submerged plant species to calculate prevalence and species diversity. We had also used a relative scoring of submerged aquatic vegetation biomass that we collected on a plant rake that had ranged from 0 to 4, where a score of 0 indicates no plants on the rake, and a score of 4 is when the rake was full of vegetation. We also collected wetland characteristics like water depth and water quality for use as additional covariates. In the lab, we had counted and identified all amphipods to the species level using up to 110x magnification. We calculated the density of amphipods and found half the study wetlands had no or very few amphipods, and per our study design, more than half had very high densities, many which had not been previously demonstrated in the literature. For example, we were finding Gammarus densities greater than 2,000 per cubic meter and Hyalella up to 7,000 per cubic meter. We used negative binomial regression modeling of Gammarus density to understand the influence of plant diversity, plant biomass, and other wetland covariates. We found Gammarus responded very strongly to high Simpson index values, but negatively with plant biomass. Gammarus were also sensitive to wetland factors like water depth, wetland size, total phosphorus, and total suspended solids. We ran a separate model for Hyalella azteca in case they responded differently than Gammarus to their environment. Like Gammarus, the Hyalella had increased greatly at high diversity values, but differently in that the densities had peaked at moderate levels of aquatic vegetation biomass. We also ran a redundancy analysis, which is a multivariate technique. In this graphical biplot, the triangles are the lakes, the blue vectors are aquatic vegetation species, all which are coded according to the USDA Plant Database species codes, and we also included 2 amphipod species as the response variables. The overall RDA model was significant, meaning that the amphipod community was well predicted by vegetation community composition. The 2 amphipod species were orthogonal, which says that the 2 species were associated with different plant assemblages. Gammarus lacustris was strongly associated with star duckweed, symbol LETR, which was a plant that was also positively correlated with Simpson Diversity Index. Hyalella azteca was associated with species assemblages comprised of sago pondweed, filamentous algae, Fry's pondweed, flatstem pondweed, and northern water milfoil. These assemblages typically had moderate levels of plant biomass, which concurred with our regression model. Kara species and sometimes coontail were dominant plants that were found in near monoculture and very high biomass, and neither amphipod species had thrived in those conditions. And lastly, both amphipod species were negatively associated with high abundance of cattails and the carnivorous plant bladderwort. So in summary, aquatic vegetation has shaped the wetland to favor high amphipod densities, and/or the loss of aquatic plant diversity could affect sensitive species. like both amphipods. Conservation strategy with focus on aquatic plant communities could result in great amphipod abundance, as well as a conservation strategy with focus on amphipods could protect wetland biodiversity. So our results suggested that these near monocultures create very high biomass, which was unfavorable to the amphipods. In monocultures, all the plants senesce about the same time of year. We think that a more diverse and staggered timing in plant senescence and the varying plant decay rates you get with plant diversity is going to provide an ongoing refuge and food base for amphipods. We suspect that we suspect aquatic vegetation diversity is indicative of overall wetland condition and promotes ecosystem functions important to amphipods like nutrient turnover and carbon availability and improved water quality. Although these so-called biodiversity ecosystem function relationships are well supported by many taxon ecosystems, these relationships are understudied for aquatic vegetation worldwide, and specifically in lakes of the Prairie Pothole region. And of course, we suspect that total plant leaf surface area and the complexity from leaf branching is going to support amphipods by providing shelter and a food source. Our results showed that restoring and managing wetlands for aquatic vegetation diversity will produce habitat that's suitable for amphipods to thrive in high densities. But although you create habitat, the question is, will they come? And we really don't know. We do know that amphipods are dispersal limited because they're unable to fly to wetlands like most macroinvertebrates can. Amphipods are restricted in their dispersal by the wings of a duck or wetlanders' waders. Wetlands with varying water levels over space and time promote predictable, diverse plant communities. Major land use changes over the past century in this region have stabilized water levels of many lakes and has adversely affected its water clarity, aquatic vegetation, and macroinvertebrate communities. Therefore, one common management strategy is to actually draw down a lake's water levels to mimic ecosystem processes that are going to kill rough fish, increase water quality, and increase aquatic vegetation diversity that could allow amphipods to flourish. A conservation strategy could also use amphipods as a flagship umbrella species to protect biodiversity at large. Both amphipods have demonstrated all the traits of a flagship umbrella species. That is, they are symbolic and charismatic in wetlands. They're easy to measure when in high density. Our results have shown they're very sensitive to the environment, particularly gamma rays. Their conservation could extend to protecting biodiversity of vegetation. And lastly, their focus could actually influence management decisions, such as whether or not to do a drawdown, or to implement a landscape mapping campaign of amphipods and plant diverse sites across the Prairie Pothole region that could inform systemic conservation planning. In conclusion, using amphipods as a flagship umbrella species can identify and prioritize wetland conservation. Likewise, the preservation and restoration of great amphipod abundance could be achieved through the explicit conservation and management of plant diversity. And with that, I'd like to thank the many funding sources, our strong project collaborators, our excellent field and laboratory technicians, and our lake managers as longtime supporters of our work. I also thank you for listening, and I would encourage you to contact me with any questions or to discuss. Thanks again.